Well, I guess I'll get started. So it is my honor and pleasure to have Oded Rechavi here from Tel Aviv. It is 11:30 p.m. his time, so be easy on him. It is late. And so he's in the Department of Neurobiology at Tel Aviv. He works on transgenerational RNA inheritance, and he's also been working recently on the Dead Sea Scrolls and doing some paleogenomics, which I find very interesting as well. And he's trying to figure out how all this memory is transmitted between generations, between mothers and daughters. And equally, Oded would probably have to be the king of academic Twitter. He has over 77,000 followers. Many people and students know him very well, particularly because of his memes. I had a lot of memes pictured here, but I can't show them, unfortunately, some of my favorites of Oded, so I'll have to show them later. But I know this will be an excellent talk, and it's an honor to have hosted you here in Madison virtually, because without Zoom, we probably would have never hosted you virtually. So have you here in person, but it is an honor having you here, and we're looking forward to hear you speak. Thank you.
Thanks a lot, Anna, for the invitation and for everything. It was I had a great virtual visit before the talk. Met some really excellent researchers and learned a lot. So it's a real pleasure. And of course, I've been following science that's coming out of your university for many years. So many things that are relevant to what I do, and such great biology. So it's a real pleasure, and I wish I could be there in person. I've never visited Madison, Wisconsin, but hopefully in the future. Okay, so today I want to talk about two projects that are both related to temperature, but they are very, very different. Both also related to memory. And one of the project will be about the effect of high temperatures on transgenerational memory. And the other part would be about low temperatures and associative memory. There are two very different projects, both connected by the link to temperature and memory. Okay, so first about transgenerational inheritance and heat. We've been working on transgenerational RNAi for a long time. I started working on this when I did my postdoc at Oliver Roberts' lab at 2010. And over the years, as you also know, probably many of you are familiar with the work of Scott Kennedy and other people in the field. Over the last decade or so, we learned a lot about transgenerational RNA in C. elegans. The first observations of heritable RNA were already in the 90s. Immediately after the Fire and Mello found that double-strand RNA is a subset for RNA, they also showed that the effect lasts in the next generation. And over the years, we learned a lot about the mechanisms and how it happens. But we now know that that aside from double-strand RNA that can trigger transgenerational silencing responses, also certain environmental conditions or stresses can lead to transgenerational changes in C. elegans. The relevance of these to other organisms is still debatable, but in C. elegans, it's very clear that not every environment, most environments don't do it, but certain environmental conditions can trigger transgenerational responses, which are probably all mediated by heritable small RNAs. The true transgenerational responses, the ones that last for multiple generations and are not just dilution of of the parental response, but the responses that are amplified and maintained for many generations appear to be all connected to heritable small RNAs. And indeed, there are different environments shown now to trigger transgenerational responses. The one that I want to talk about today is temperature. This is a partial list, unfortunately, of all the temperature-dependent transgenerational reports. There are others. For example, we know that the mortal germline phenotype, the fact that certain mutants of C. elegans that are defective in epigenetic processes in production of small RNAs, and also in production or in certain histone functions, become progressively sterile over generations, and this is accelerated in high temperatures. But there are also other reports showing that exposure to temperature leaves a transient, generates a transient effect that affects the physiology of the worms and affect many different characteristics in the descendants. And this is a partial list. So how does it happen? How is RNAi inherited over generations? I will not show you too much on that, just a little bit. We now know that the small RNAs are carried transgenerationally in the germline by dedicated argonauts. There are 4 argonauts that have been implicated specifically with transgenerational carryover of small RNAs. One of them, a very important one, is HRD1. It's also known as WAGO9. This is from Scott Kennedy's paper, where he showed that HRD1 mutants, HRD1 stands for heritable RNA deficient 1, they do RNA just fine in the parental generation, but the RNA response is not inherited to the next generation if this argonaut that carries small RNAs in the germline is not there. The small RNAs that can get inherited because they get amplified in every generation. Otherwise, the response would immediately dilute because worms produce hundreds of progeny. So you have to amplify the response to maintain it over multiple generations. And in C. elegans, different type of small RNAs act as primary small RNAs that do not lead directly to silencing. So for example, endogenous siRNAs or exogenous siRNAs or piRNAs or also ribosomally associated small RNAs. All these piRNAs do not lead directly to silencing. What they do is they recruit to the mRNA template RNA-dependent RNA polymerases, which use the mRNA of the target as a template to synthesize much more abundant secondary small RNAs, which are the small RNAs that will get inherited across generations. And these small RNAs have a typical length and an end. They are mostly 22 nucleotides long, and they start with a G. And recently, Scott Kennedy's lab has shown a very interesting feedback where the RNA that gets targeted for degradation is modified, and there's an addition of U's and G's that are added in a non-templated manner to the end of the RNA, and this allows recruitment of the RDRPs for additional rounds of amplifications, a sort of a cycle that gets created. While the worms can, in theory, maintain this cycle forever and create more and more small RNAs in every generation, we know that the response eventually stops. They typically stop after a few generations. When it comes to exogenous RNA, the response normally lasts for 3 to 5 generations, and then it stops. It doesn't stop because of dilution of the original RNAs that are made in the parents, because they are amplified in every generation, but instead it stops because there are mechanisms that terminate it after a few generations. And we, in 2016, called it the transgenerational timer. These feedbacks between different processes that lead to the resetting or that stop transgenerational RNA responses. And we identified genes that we called MOTEC genes. This stands for Modified Transgenerational Epigenetic Kinetics. MOTEC, to the Hebrew speakers in the audience, means sweetheart in Hebrew. And also later, Kennedy also found the genes that he called hairy. It's also determined the duration of the heritable response. And there are different mechanisms. For example, we've shown that production of endogenous small RNAs of different sorts comes at the expense of exogenous RNA responses. The reproduction of endogenous small RNAs eventually leads to termination of the exogenous RNA responses, in part because these endogenous small RNAs regulate the RNA inheritance machinery itself. We also showed that certain chromatin modifiers like MET2, which is a H3K9 methyltransferase, function in terminating heritable RNA responses. And in MET2 mutants, RNA responses last essentially indefinitely. We now also know that stress can reset heritable RNA responses. So as I told you, if you start an RNA response by challenging the worm with double-strand RNA, typically it would last for 3 to 5 generations. However, we found that if you stress the F1s in a number of different ways, you can starve them or put them in high temperatures or put them in high salt, then the heritable response would stop immediately at the F1s. So the response is plastic and stress can reset it. Now, the link to temperature is very interesting. Temperature generates transgenerational response. It can also terminate transgenerational responses. And last year, we've shown that HSF1, this master regulator of temperature response, heat shock factor 1, generates that stochastic activity of HSF1 generates variability between different isogenic worms, sisters, variability that determines the fate of the inheritance that each of the sisters will initiate. So while the worms are isogenic, if you take one particular mother and you look at its progeny, while the worms are isogenic, they're all sisters, almost genetically identical, each of the sisters will transmit to its progeny responses that are very different from the responses that the other sisters would transmit. And this is largely dictated by stochastic activity of HSF1, which regulates many factors that control RNA inheritance. And that the HSF1 status of the sisters, of the daughters, dictates or determines the fate of the inheritance in the progeny. So again, this is a very interesting link to temperature. We also found that RNA mutants that appear to be normal have some abnormalities when you look at slightly higher temperatures. So for example, RD4, which is a double-strand RNA binding protein that acts relatively upstream in the production of different small RNAs, the worms look normal, and for many people thought that they don't have any striking phenotype of effect aside from the defect in RNA. We found that they are defective in chemotaxis in high temperatures, in 25 degrees. And 25 degrees is slightly higher than the normal temperature that we grow worms in, but it's still a normal temperature that the worms can reproduce and they're fine. It's a mild stress. But you can see here that the worms don't do chemotaxis well, that RD4 mutants don't do chemotaxis well in high temperatures. And you can also see the tracks. This is a worm tracker. These are tracks of N2 worms, which are the wild types, or RD4 mutants, these RNA mutants. And you can see that the RD4 worm doesn't forage as much as the N2. It doesn't explore. It stays in the middle of the plate. It's easier to see if you look at many worms. Now you see that the RD4 worms in high temperatures only, they stay in the middle of the plate and they don't explore much. And we wanted to see what's wrong in these worms in this high temperature. And this was published, so I won't get into it, but the bottom line is that we found that small RNAs that are made by RD4 in the nervous system generate transgenerational responses, eventually changing the pool of heritable small RNAs in the germline for multiple generations, and that some of these small RNAs control the expression of a gene that's called SAGE2 that you must downregulate for the worm to find food properly, to do chemotaxis properly in high temperatures. And we've shown that this is an HRD1 dependent phenomenon. So it depends on this argonaut that transmits small RNAs across generations. And there are still many mysteries left here of how this exactly works. And I saw that some of the students had questions about it that we are currently exploring. But the bottom line that I wanted you to get now is that temperature generates transgenerational responses, and also the temperature dependent behaviors are affected by heritable small RNAs. So temperature is in the mix of all of this. In our paper, and also in other papers about RNA inheritance, it becomes clear that small RNAs can move from the soma to the germline. And this is a very interesting thing, because one of the basic principles of biology is that the germline should be segregated from the soma so that somatic responses cannot transmit to the next generation. This is called the Weismann barrier, or the second law of biology. And it's one of the barriers that should stand in the way of inheritance of acquired traits. In C. elegans, it's obviously breached this barrier. and RNA responses can move from the soma to the germline. However, we found, and also other labs have shown, that it appears that in most cases, the genes stay silent, the target stays silent in the germline, but the response doesn't move out of the germline back to the soma. So it's a sort of a reverse Weismann barrier. So for example, if you have a GFP expressing worm and you treat it with RNAi, in the first generation, GFP will be silenced all over the worm, but in the next generation, it will only be silenced in the germline, and GFP will be re-expressed in the soma. The RNA responses don't go back from the germline to the soma. So you might ask yourself, how can you affect, for example, by temperature or by neuronal small RNAs, how can you affect behavior in the next generation if the silencing only goes one way, from the soma to the germline? Shouldn't you transfer the response back from the germline to the nervous system? The answer is it's not a must, because the germline communicates with the soma in many different ways. Many molecules are secreted, and we know that there are works in C. elegans showing that chemotaxis behavior is disrupted if you ablate the germline, and that also specifically temperature-dependent behaviors are affected if you just ablate the germline. This is because the germline affects the soma in many ways by secreting different molecules, and also because the germline controls or affects the dynamics of the development of the soma. So I think you can affect somatic functions in the next generations indirectly by heritable small RNAs that remain in the germline, that only regulate germline genes. I would also add that there are some exceptions to the rule, and some genes can get silenced in the soma transgenerationally, but this is a very interesting exception, and we are trying to understand what's going on there. Okay, so we asked ourselves, can transgenerational responses affect the process of evolution by affecting behavior when we know that they are mostly restricted to regulation of germline genes? It's a sort of a complicated hypothesis. The reason that we even ask this is that I'm very interested in evolution, and I'm always thinking, can small RNA inheritance, can this unusual form of inheritance, which enables inheritance of parental responses and some acquired traits heretically to transmit between generations, can it affect the process of evolution? The initial thought is that it can't, because most of the responses are short-lasting. You start an RNA response and it lasts for a few generations, but then it's gone. And mostly when you think about evolution, you think of a process that takes many, many generations to occur. So how can a transient effect affect a very long-lasting process? And we thought that perhaps it can happen by, if small RNAs can affect behavior, because if you affect behavior, you can carve new niches that would affect evolution indirectly. But to affect evolution, I remind you, the small RNAs must do it by affecting the germ cells. So these were all the restrictions that we had when we started this project. Okay. Our starting point was that worms have two very crucial modes, two very different modes of reproduction. They can self, because the worms are hermaphrodites, they make both sperm and egg, so they can self-fertilize, or they can outcross with males, which are very rare in the population. And this is a very dramatic decision, because mating, when you mate, you expend a lot of energy, you take a big risk, because you can get eaten, you risk predation, and also importantly, when you mate, you dilute your genome in half, because you only contribute half of your genetic material. When you self, you basically replicate yourself, okay? So the worms, when they choose to mate with males, it's a very big decision. And nevertheless, it's clear that outcrossing has advantages as well. In many different organisms, and also in worms, and specifically in C. elegans, it has been shown that under unpredictable environments or under stress, it's worth it to outcross, because indeed you dilute your genome, but when you outcross, you extend genetic diversity. So you give evolution more raw material to select from. And this could maybe lead to, you can maybe gain beneficial alleles that will allow you to survive. It's worth it to increase the diversity, genomic diversity. So we wondered whether small RNA inheritance can affect or can control this decision of selfing versus outcrossing, because if it could, then it could affect genetic diversity, and therefore even a short-term effect might affect the process of evolution. Okay, evolution according to the basic selection according to the environment according to the basic theory or the classic theory of evolution should affect selection. The environment should affect selection, but not variation. Okay, even stress-induced mutagenesis, even in bacteria, is still very controversial. Okay, now the worms can choose whether to mate or to self. I don't want to give them free will, but so I won't say choose. I say they can affect their propensity to mate with males by secreting a pheromone that attracts the males. And this has been shown by a number of labs, by Sternberg's lab, Maureen Barr's lab, and the Chu lab. So they didn't identify the pheromone yet, but what they found is that worms normally do not secrete this male attracting pheromone. So when they self, they only secrete the male attracting pheromone when they become old. The reason is, so if you take, if you extract odors and pheromones from young adults, from young worms or from young adult worms, these odors that you extract are not attractive to males. However, if you extract odors from old worms, the males will be attracted to them. Okay, in a simple chemotaxis assay. And the reason is that the worms only make sperm when they are young. And then they run out of the hermaphrodites. And then they run out of self-made sperm. And if they want to continue to reproduce, they have to secrete a pheromone and get males to inseminate them to supply new sperm so that they can continue to reproduce. Okay, so they only secrete the pheromone when they are old. However, we knew, and the way that they do it, it's not entirely clear, but the way that they do it is that there's some mechanism, hasn't been identified yet, that the oocyte sensed when it stops being fertilized by sperm. When the sperm stops being made and the oocyte doesn't get fertilized, then the pheromone is secreted. Now, we knew that small RNAs, including small RNAs that are affected by different environments, regulate many genes in the germline and can control fertility. So we thought that perhaps under stress, these small RNAs will be disrupted, leading to disruption in the production of sperm, and then the oocyte will sense this disturbance in the force and will start secreting the pheromones, and perhaps this would lead young adults to be attractive too. They will also secrete pheromones and mate with males. This was our hypothesis, that perhaps environmental challenges can change sexual attractiveness and the likelihood of crossing by affecting small RNAs in the germline. I'm skipping a lot, but what we did is we looked at many different environmental stresses. Most of the stresses did not make the worms attractive when they were young. They remained unattractive. They didn't secrete the pheromone. However, when we grew worms for a few generations in high temperatures, in 25 degrees, they started being attractive even when they were young, prematurely attractive. And this attractiveness depends fully on SRD1, which is a pheromone receptor identified by the Chu lab that is expressed in the AWA neurons of the males. So now we know it's the same mechanism that has been studied by the Sternberg labs and Maureen Barr's lab and the Chu lab. Okay, the worms secrete the same pheromone that they were studying in the past. The pheromone hasn't been identified yet. As a positive control for worms that are always attractive, we use FOG2 mutants. FOG2 mutants don't make sperm, so they are always attractive, even when they are young. Okay, so we found that when you grow worms for one generation in 25 degrees, they are not more attractive. but they are not attractive when they're young. But when you grow them for 10 generations, they become attractive at 25. When you grow them for 10 generations at 25, they become attractive. Okay. Being attractive is one thing, and mating more is another thing. Okay, it's more complex. Mating depends on many factors. So we wanted to see whether the worms that are grown in high temperatures are also mate more with males. So we let the worms that were grown in high temperature compete with worms that were grown in normal temperature in the same plate over males that have fluorescent sperm. Okay, so we can follow insemination. And we can distinguish between the worms that were grown in high temperatures and the worms that were grown in normal temperatures because they have different fluorophores. And so we let them compete over males, we remove the males, and we look for insemination with a fluorescent sperm. And what we saw is that the worms that were grown in high temperatures for a few generations, not only do they secrete more pheromone, they also mate a lot more than the competitors, the worms that were grown in normal temperatures. But is it inherited? Of course, this is the most interesting part. And the answer is that it is. It is inherited for 3 generations, and then it goes away, like many other responses that we see in C. elegans. So we grow the worms in high temperatures, we shift them to 20 degrees, normal temperatures, and they remain attractive for 3 generations robustly, and then it goes away. And the fact that it goes away tells us that it's an epigenetic effect. It's not because of some change in the genome, for example, jumping of transposons in high temperatures. Even if transposons were to jump in high temperatures, this wouldn't lead consistently always to the same phenotype. But here we know that it's a transit epigenetic effect because it goes away after 3 generations. The next question is, of course, how is the response transmitted across generations? Is it another small RNA mediated effect? And to check this, what we normally do is we examine the same effect in mutants that can't inherit RNA. And our mutants of choice are HRD1 mutants. This is the Argonaut that carries small RNAs across generation, which is needed for most, but not all of the transgenerational RNA responses that people studied. The problem is that HRD1 mutants are temperature sensitive, so we can't use them for these assays. So what we did is we tagged the endogenous HRD1 protein, the Argonaut, with the degron, and we expressed just in the germline tier 1, which is the enzyme that degrades the degron-tagged protein. And this is an inducible system. When you add auxin, this plant hormone, you induce the degradation of the degron-tagged protein, in this case, HRD1, the Argonaut. So what we did, we grew the worms in high temperatures, and then in the next generation, we depleted the HRD1. So we led to a reset of small RNA inheritance. And what we saw is that the worms stopped being attractive. This stops transgenerational attractiveness, which means that the heritable attractiveness is because of heritable small RNAs that are carried by HRD1. We have many additional controls for this, just tier 1, just the degron-tagged protein, and so on. Okay. We wanted to know which small RNAs do this, which small RNAs get inherited to mediate the effect. So we screened through many mutants that function in the production of different small RNAs species. And we found 4 different mutants that are prematurely attractive, that secrete the pheromone already when they are young, and also that mate more with males. However, only one of them, in fact, it's a double mutant, MEG3, MEG4, also transmitted the attractiveness to wild-type descendants. Only these mutants led to a transgenerational response. I will tell you what MEG3-4 are in a second. But you can see here, we start with MEG3-4 mutants, homozygous mutants. They are prematurely attractive. We cross them to males, to wild-type males, and then we isolate HEF2 homozygous wild types. And we see that they remain attractive, and also their descendants for multiple generations, this lasts for more than 3 generations, remain attractive. And this transgenerational attractiveness that results from disruption of MEG3-4 depends fully on HRD1. So while HRD1 mutants on their own are not prematurely attractive, HRD1 resets the transgenerational attractiveness that is initiated by disruption of MEG3/4, because HRD1 carries the small RNAs transgenerational. So you see that the triple mutant is not transgenerationally attractive at all. What are MEG3/4? So they are core germ granule proteins that are important for organizing these liquid-like condensates that reside in the C. elegans cytoplasm of the germ granule outside of the nuclear pore. And in these granules, many of the factors that are important for RNAi and RNAi inheritance reside. And we've shown, and later also, just after our paper, a month later, also Scott Kennedy's paper and Geraldine Sedou's Scott Kennedy's lab and Geraldine Sedou lab showed the same thing. We found that if you disrupt the germ granules by mutating MEG3 and 4, or by mutating other germ granules proteins, this leads to transgenerational changes in RNAi that are very, very robust and lasts for multiple generations. They also show interesting parent of origin effect. It matters whether you disrupt the granules in the paternal or maternal lineages. But if you disrupt these, if you look at these mutants and they're also in the wild type descendants, the composition of the heritable small RNA pool is totally different, and this is very, very robust and lasts for multiple generations. So what we think is that when we disrupt MEG3/4, this disrupted germ granules, this leads to transgenerational disruption of heritable small RNAs, leading to the attractiveness. And we found that this is actually validation of stuff that other people have shown too, is that the granules, these germ granules, change in high temperatures. There are a few different granules. Again, Scott Kennedy, who discovered so many things in this field, found that there are germ granules and there are also Z granules that bud off the P granules. We found that the P granules, but not the Z granules, shrink in 25 degrees. So we think that temperatures can affect the characteristics of the granules and therefore affect transgenerational inheritance via the granules, leading to sterility. We wanted to know whether, in fact, there are problems in the sperm of MEG3/4 worms and also in the wild type descendants. So first of all, we characterized their brood size. We saw that MEG3/4 mutants have a smaller brood size, but we found that this is partly because of specific problems in the sperm, because if we supplement, if we cross them to wild type worms that supplement wild type sperm, this largely solves the problem in the brood size. So they have specific problems in production of sperm. So it all fits together. And also when we looked at the small RNAs that are disrupted transgenerationally in descendants of MEG3/4 mutants, and also in worms that derive from ancestors that were growing in high temperatures, we saw that among the small RNAs that get transgenerationally changed, there's an enrichment in small RNAs that target sperm genes and also changes its sperm mRNAs. So when you disrupt the germ granules or when you grow the worms in high temperatures, this leads to transgenerational changes in sperm functions and also in small RNAs that target sperm genes and regulate their activity. Okay. So we think we have some of the mechanism, but we wanted to get, but the question that we started with is, can these transgenerational effects eventually change evolution, the process of evolution, or can this transit epigenetic effect change the permanent genetic composition of the population? Can an epigenetic effect indirectly, because of increased mating, change the composition of the genetic composition of the population? And to ask this, to show this, what you need to do is to do lab evolution experiments. And of course, lab evolution experiments are typically done with single cell organisms because they reproduce so fast, but you can also do them with worms. The generation time is 3 days, so it's doable. But of course, you can't do a million generations. You can just do a few. So what we did is we took two competitors, groups of worms that are genetically identical. However, they have different epigenetic lineages. One of the groups come from wild type ancestors, and the other group descends from ancestors that were mutated for MEG3/4. So we know that they are transgenerationally more attractive. We can distinguish between them because the ones that the epigenetically attractive lineage is GFP positive. Okay, they express GFP. But now they are genetically identical, and we let them compete over males. But we do it for multiple generations. We keep moving the worms between plates and adding males, and we quantify and we count the proportion of GFP positive animals versus GFP negative animals. And we picture all the worms on the plate, and we analyze the proportion of the worms using an automated software that measures fluorescence that we called War Machine, that we published a few years ago. This is a machine learning based algorithm, software that you are welcome to use. And we do this big lab evolution competition experiments in two ways, either using wild type worms or using worms that can't sense the pheromone because they don't have the pheromone receptor, SRD1. So the advantage of secreting pheromone and being transgenerationally more attractive is canceled. And what we found that only when the males can sense the pheromone, there's an advantage of being transgenerationally attractive, and the epigenetically attractive lineage that descends from NEGFI4 ancestors wins. It spreads its alleles more efficiently in the populations and becomes more abundant. It takes over. So here we see that the process of evolution is affected indirectly because of accelerated mating or more mating because of epigenetic inheritance. So to summarize everything, what we found is that high temperature disrupt the germ granules, affecting smaller RNA inheritance and targeting of sperm genes. And when the sperm doesn't function optimally, the worms are still fertile, but less so, then the oocyte senses it somehow. There's a mechanism that senses it, and the worm starts secreting a pheromone that attracts the males, encourages mating, and eventually leads to diversification of the population genetically. Okay, this is the first story that I wanted to tell you. The second story is already also related to memory, but it's very, very different. Because here I will be telling you about low temperatures and about memories that are kept within the same generation. Associations that the worms acquire using its nervous system and keeps throughout its lifetime, not transgenerationally. Now, while the worms are very good, they're a great model for studying transgenerational inheritance, and they have transgenerational memories that last multiple generations and months. When it comes to memories that they remember within the same generation in their lifetime, they can only acquire very short memories. And this is really disappointing. In fact, the reason that I wanted to study C. elegans is that it has such a simple nervous system, just 302 neurons. So potentially you can understand how memory is encoded in the brain. That was what attracted me originally to go to study C. elegans. Okay. However, people have shown, and this starts in the 80s with works from, or 90s, early 90s, with works from Corey Bergman and Horowitz and others, is that if you teach the worm something, typically they will forget after just 2 hours. Very disappointing, okay? And the question is whether, and you can still do a lot and study a lot, learn about it, and learn a lot about it, and there are exceptions, but by and large, this is the rule, and it is disappointing because it's a short-term memory. It doesn't last for a long time, okay? The question is it a bug or a feature? When we think about memory loss, we think about old age, or we can't remember what we need to remember for a test. So we think of it as something bad, but it's also something adaptive and good, because if you remember everything that you ever learned, you will be completely post-traumatic, and also you'll remember many irrelevant things that you no longer need to remember, right? So the question is it because of the worm's environment, or perhaps the type of memories that or the type of learning essay that we use? Perhaps there are some things that the worms can remember throughout its entire lifetime, or for more than 2 hours. Okay? That's a very interesting question that I wanted to study, but as a start, what I wanted to do is I wanted to take worms. It was a very naive idea. What I wanted to do is, you can freeze worms. You can freeze worms, and every C. elegans does it. Every C. elegans lab does it. We freeze worms all the time, and we have stocks of frozen worms. So what I wanted to do is I wanted to teach the worms something, and then I wanted to freeze them, and you can keep them in -80 forever pretty much. And then I wanted to thaw the worms and see whether they still maintain the memory. They are incapable, appear to be incapable of long-term memory, but maybe you can freeze their memories artificially, because you can freeze worms quite efficiently, okay? That was the naive idea that I had. And the reason that I had such a weird idea is that many theories about how memory is encoded say that memory maintenance depends on electrical activity. And when the worms are frozen in -80, there isn't any electrical activity. So I thought if the memories will last after freezing, this would mean that electrical activity is not important, is not necessary for maintaining of memories. That was the motivation, okay? The problem is that you can only efficiently freeze worms when they are L1 larvae. The worms transition through 4 larval stages, and they can only be efficiently frozen when they are small. Probably because when they are big, the ice crystals break the cells. And most of the learning and memory assays that are good and robust are with adults. So it's a problem. It's just a technical problem. So I gave a few students in the lab the challenge of trying to freeze adults and asked them to do this experiment. No one agreed until one student, Dana Landschaft, agreed to do this experiment. I was very happy that I have such a brave student. Little did I know that she would just ignore me and do something completely else that I didn't ask her to do. What Dana did, and I'm very happy that she did it in hindsight because this led to a discovery and I wouldn't think of doing this, is that instead of freezing the worms in -80, she taught the worms an association and then she just placed them on ice. So they're not frozen, they're kept at around 0 degrees, okay? But they're not frozen. Cooled down, certainly, but not frozen, okay? But this is what she did. She wanted to see how temperature affects memory acquisition or maintenance. And the most robust assay in C. elegans for learning and memory is this memory. You take an appetitive odor that the worms innately love, they are attracted to it, and you pair it with starvation. And then the worms don't like this odor anymore, and they don't go towards it. So it loses its appeal. It's not attractive anymore. And this is the type of one of these memories that only lasts for 2 hours, okay? Done by many labs. So what Dana did was she took worms and she starved them in the presence or in the absence of this innately attractive odor. In this case, here in the first experiments, I think she did with butanol. It's a chemical, an odorant that is attractive to the worms. And then immediately after doing this learning paradigm, she tests whether it worked, whether the worms learned by doing a chemotaxis assay. And as I'll show you in a second, she sees that immediately after the training, the worms are no longer, only the worms where the odor was paired to starvation are no longer attracted to the odor, okay? But in addition to doing this, to immediately test them, she also keeps other worms, she let them wait for a few hours to see whether the memories will be forgotten. And she let them wait either in room temperature or on ice when they are cooled down. And then after a few hours, when the worms should already forget, she tests them, she does another chemotaxis assay to see whether they remember the memory or forgot it, okay? This is the assay that Dana invented. And what Dana found is extremely surprising. She found, and for me it was shocking in the beginning, that when the worms are kept on ice, they don't forget. Okay, they maintain the memory for as long as they're on ice. So you see here, this is the chemotaxis assay of worms. Immediately after the training, you can see that the chemotaxis assay drops because immediately after the pairing of the odor with starvation, they are no longer attracted to the odor. However, after 2 hours, they forget. The chemotaxis index is again high. The worms move towards the odor. They forgot the association. However, worms that are kept on ice after 2 hours do not forget, okay? They are still unattracted to the odor. And I think it's easier to look not at the chemotaxis index, but at the learning index, which is just the chemotaxis index of the naive minus the chemotaxis index of the condition. It's just more intuitive. You can see, and this is a measure for whether they still remember. So you see that immediately after the training, the learning index is high, but after 2 hours, they forget. So the learning index drops. But if the worms are on ice, the learning index is still high. They don't forget, okay? And this doesn't last only for 2 hours. Even after 16 hours, the worms on ice do not forget. We also did experiments where we wait for 24 hours. They still remember. However, at that point, they become sick and half of them die. So we can't do these long essays. The worms remember on ice not because the neurons lost the capacity to smell. This is not the reason that they are no longer attracted to the memory, the butanol, because they are still attracted to other odors like benzaldehyde, which is also sensed by the same neuron. Okay, so they are so the same. The neuron functions. It can sense one odor, but it's not attractive to the other odor that was paired to starvation. The memory, the worms don't forget while they're on ice. However, the memory is not consolidated because as soon as you remove the worms from the ice, the clock starts ticking and forgetting begins. And after a few hours, they will again forget. So putting the worms on ice just delays forgetting, but but the memory is not consolidated. Now, we originally thought that what happens is that temperature just delays whatever forgetting is. So every reaction slows down in low temperature, and perhaps the processes that leads to forgetting are just delayed at low temperatures. The kinetics of the enzymes or something is slower, so they forget slower, okay? But we no longer think this is the case. In fact, we are sure this is not the case because of a number of reasons, okay? I will show you the most convincing one. There have been beautiful studies conducted over the last 20 years from different groups which showed that worms can become cold tolerant, okay? If you take worms and you place them, if you take C. elegans and you place them on ice for 48 hours, for a long duration, then they all die. However, if you first acclimate them to slightly lower temperature, so instead of growing them at 20 degrees, you grow them at 15 degrees overnight, or you can do it for a minimum of 5 hours, the worms change an internal state and become cold tolerant. Now, if you place them on ice, they will all survive, okay? And this is because of changes in lipid composition. It is also supervised or orchestrated by the nervous system. And these groups have done a lot of excellent work identifying genes that are required for cold tolerance. We wanted to know whether there's a link between cold tolerance and the capacity of worms to remember longer on ice. So what we did is we repeated these experiments, only this time, before we taught the worms the association between the odor and the learning, we acclimated them to lower temperature. We made them cold tolerant. And what we found is that when worms are made cold tolerant, they no longer remember, they no longer maintain memories on ice. You see the learning index here, when the worms are cultivated overnight at 15 degrees, and then you teach them, and then you place them on ice, the memory is not extended. So when they switch to this internal state of being cold tolerant, memory is not extended on ice, which means that it's not just the temperature that controls the memory. It's not just a slowing down of metabolism or slowing down of enzymatic reactions. It's this internal switch that changes, okay? And this is plastic. If you grow the worms, if you make them cold tolerant by growing them overnight in low temperatures and then grow them in normal temperatures for 2 hours, they switch the internal state back to being cold sensitive, and then memory is maintained on ice. Okay, so this is something very plastic. And also cold tolerant is plastic in a similar way. And this was a great way to get at the mechanism. What we did is we sequenced mRNAs from worms on ice after they were taught the association or not. But we in 3 different groups, we sequenced mRNAs from worms that are cold tolerant, cold sensitive, or worms that were made cold tolerant and then made cold sensitive again. And we looked at the genes that change when the worms are on ice. And we found genes that are required, that were shown in the past to be required for cold tolerance, that are also needed, completely needed for maintaining memories on ice. For example, the entire pathway that leads to synthesis of deacylglycerol. If you disrupt this pathway in any number of ways, the worms don't maintain memory anymore on ice. This is very interesting because also deacylglycerol is controlled synaptic release. So this could be the memory actually. But more interestingly, we wanted to see whether there are genes that when you disrupt them, the worms remember better, on ice or off ice. And one of the genes that change in this mRNA sequencing is TRX1, which is the worm's thyrodoxine, which caught our attention because it is expressed just in a single pair of neurons called ASJ. The reason that it caught our attention is that ASJ neurons were shown in the past to be important for cold tolerance. Okay? They coordinate cold tolerance. So we checked TRX1 mutants and we found that they maintain memories longer. You see, we didn't check for 16 hours, but even after 5 hours, they don't forget. Normal worms forget after 2 hours. And this is happening even when the worms are off ice. So they never experience the low temperatures. They just have longer memories if they are mutants. Not only that, low temperatures does the opposite. It shortens the memory maintenance of TRX1 mutants, which is very weird. To tell you the truth, this is the result we had in the beginning, and it is very consistent when you examine this mutant of TRX1. However, we found very confusing results when we check other mutants of TRX1. So we don't really understand what's going on yet. And we are figuring this out by checking different mutants, many different mutants of TRX1 and also their interactions with other mutants. However, I remind you that TRX1 caught our attention because it is expressed specifically in these ASJ neurons, which are required also for cold tolerance. So we wanted to know whether there's a way to affect ASJ function, whether we can perhaps even use a drug to affect ASJ to extend memory. And we didn't know how to do it. We had no clue how to do it. And then what happened is that we got lucky. So we all went for a retreat to the desert where it's very hot, not cold. And on the way back from the retreat, we listened in the car to a podcast. It was a Radio Lab podcast about lithium, which is a very, very interesting drug given to people with bipolar disorder. And it's interesting, not only because it's a wonder drug of psychiatry, very potent, but also because lithium is just an atom, the third atom in the periodic table, made in the Big Bang. You can find it naturally in salt flats, for example, in Bolivia. And although it's just an atom, it's an amazing drug that affects our psychology. So while I was driving in the car listening to the podcast, I thought this was completely unrelated to the stories that you just heard. I thought maybe we can test lithium, how it works on C. elegans, because C. elegans has such a simple nervous system. Maybe we can understand how lithium works because no one really knows. Although this has been given as this is a drug for many years, no one knows how it works. Maybe we can study it in C. elegans. So I listened to the podcast, and as soon as I stopped the car, I got home, I Googled it, and I saw that it's not an original idea. In fact, people studied it, and what they found is that lithium works in C. elegans specifically on the ASJ neurons. Not only that, in this paper from Dennis Kim's lab, they found that it works specifically on ASJ by working on this phosphatase, BPNT1, that acts on a target that's just in ASJ neurons. And they have a figure in the paper where they show that TRX1, this gene that we find to be involved in memory maintenance, is downregulated. Do you see? So I immediately told Dana, you have to study to throw some lithium on the worm and see whether it affects memory. And what Dana found, that this has been repeated now for many times, is that worms that are grown with lithium overnight have a longer memory. They don't forget this association. Even after 5 hours, they don't forget. And we're now studying whether this is connected to cold tolerance and whether this really happens via the ASJ neurons and perhaps TRX1. There are still many unknowns here, but I'm showing you preliminary results that are still unpublished. I think this is the most exciting thing. So I will stop here. I hope I didn't go over time, and just thank everyone that was involved, especially the students who actually did the work. So Dana, who led this second story, and Itai, Yael, and Itamar, who led the first story, and also our collaborators in the Weizmann Institute, Rockefeller University, and Tel Aviv University. And you for listening. Thank you very much. And of course, I'll be very happy to discuss all of this and answer any question. Thank you.
Thank you, Oded. Does anyone have questions?
Hey, can I ask a question?
Yeah.
Sure. Hi, thank you so much. That's very interesting. I have a question in the second part. So, so you meant that the memory in the C. elegans is that stored in the one single cell to maintain in the short term, or multiple?
So what we know is that the activity of this single cell, in fact, two cells, AWC, which is a sensory neuron, and SJ, which appears to be involved, that these two cells affect memory maintenance. Where exactly is the memory maintained, or what is the memory? That's a big question. One possibility is that indeed it is diacylglycerol affecting synaptic release. But exactly what is the memory? It's a bigger question that we didn't fully answer.
Okay. That's interesting. Is it easy to know in the C. elegans how many cells are involved in the memory formation, or is that testable? Just knocking down some of those. I don't know. I'm not using the C. elegans as a model.
So I think it's possible. I think we can narrow it down eventually to understand what is the memory. This is the goal. This is what we're striving to do. And indeed, if it's possible in any organism, it's probably in C. elegans, which is a very simple nervous system.
That's interesting. Thank you.
Thank you.
Any other questions? Please speak up. Any more questions?
Hi,
Hans?
yeah, I have a quick question. So the first part, so is how does the generation time of the 25-degree actually, why 10 generations the magic number? Or how, I mean, the decaying of the effect have about 3 to 5, there's a clock about that. How about the accumulation? Is that 10 is the magic number, or maybe 5, or is a certain number, and how is that affected?
Yeah, that's a great question. So indeed, we and also others found that there are genes that affect the duration of the memory, transgenerational. But you're right that there are two things here. There's the accumulation of the memory, of that you need to, something accumulates in 25 degrees. And there are also the counting of 3 to 5 generations. In the MECP4 mutants, it lasts even longer. We don't know exactly how the worm counts and what is it that accumulates. It's a very, very difficult question, and we don't know. It could be an accumulated damage of disruption of the germ granule functions or sperm functions, but exactly how the worm counts generations is unclear, and one of the big questions.
Cool, thanks.
Hi, Oded. I had another question. I was wondering if you tried different concentrations of lithium chloride and seeing if that affected the duration of the memory.
We didn't do this yet. You know, there are so many things we could do, and this is a great idea. But this is so slow. Dana can only do 2 experiments a week. So there are a million experiments I want to do, but we didn't do it yet. But it's a good idea. Yeah.
I had a question about the memory. So this would be short-term memory that the worms are learning?
It depends. It depends. I don't think it depends on protein translation on translation, though we didn't check it. So that's one definition of one way to differentiate long from short. It's not a memory. It's not a short-term memory of seconds. It's something that has a few hours, but it's a good question of whether it depends on translation or not. That's one way to distinguish short from long memories.
Because I was just wondering if, I was just wondering if there's certain, like if you get a concussion, it can affect your short-term memory, the memories that you made right before. And I don't know how you would test this in worms, but how might ice affect the loss of, would it preserve their memories even after concussion, or maybe it doesn't, maybe it operates through a completely different pathway?
I'm sorry, I didn't fully understand. Can you repeat? Sorry.
What I was wondering is, so the being on ice preserves their memory longer.
Mm-hmm.
We know in humans that concussions can wipe out your short-term memory from right before the accident. So
Right.
if the worm got a concussion but was on ice, would that preserve their short-term memory?
Yeah, so that's a very interesting question. I don't know about concussions in worms, but I do know that there are some protocols for preservation of memory after all kinds of things that you lower temperatures. You lower the person's temperature. You also do it in anesthesia. Part of the procedure is cooling the person down. And it's true that there's loss of short-term memory. However, there are also reports about that you forget what you were taught immediately before, but you do remember longer even things that you learned a little bit beforehand. So we talk with people who do anesthesia, and this is what they told us. But of course, the gap between worms and humans is very big, and the links are only speculative.
Great. Anyone else?
Hi, I have a couple of maybe facetious questions. One is for the short-term memory, do you think it's going to work on the human population? I'm just thinking of my big class of undergrads. Should I teach them something and have them go into the cold room? Are they going to retain it more? And just on the same lines, what do you think this has to, what about interstellar travel? Does this really say that we should, teach the astronauts something and do, send them cryogenically?
Yeah, so of course I can't really say. We have no idea. I should put a disclaimer on everything. I normally say it in the question and answer thing that everything we find in worms, we hope will be somehow relevant to other organisms, but we don't know that's the case. Okay, so first of all, important disclaimer to say. I always follow this up by saying that even if it's only in worms, it's interesting to me. And we should remember that 4 out of 5 animals on this planet are worms. So we are the exceptions, not them. Okay, so it's important to study worms on their own. But so I just don't know. I have no idea. I can tell you that people that are used to low temperatures have changes, long-lasting changes in their physiology. Just maybe a month or 2 ago, there was a paper, I think in Neuron, about Finnish or Danish swimmers that swim in ice temperatures, and the many characteristics are changed. Many things in the physiology are changed for a very long time. So obviously it has an effect. Whether it affects memory, I have no idea. But I think many things are slowed down in low temperatures. In our case, in the worms case, I remind you, it's not only because of slowing down of the physics. It's because the worm switches an internal state that somehow is more conductive to maintaining memories on us. It's just not just a slowing down of every reaction. But I don't know. I like how this is improving. First we had cats, now with kids. I wonder what will be next. It's getting better. Yeah.
Is that the last question? We'll let Oded give everyone applause. Thank you for coming. And
Thank you.
Send you off to the class. So Jerry and Aki will take over. Thank you, Oded.
Thank you.
Well, there's something in the there's a thank you in the chat. All right, I'll see you later.
See you.
Sorry. Sorry, I had to listen to on the phone. My computer died. In trouble. So
You should send your slides to everybody for the introduction.
my favorite memes from Oded, I'll send it out to the department. So
That's good.
all right, have a good class.
Hey, thank you, Anna.
Bye.
Back in a second, okay, just 1 second.
Oh, sure, no problem.
Okay, now I wanted to see whether I have wine or beer or something, but I don't have anything. So it's okay.
Well, I see that you are already past the midnight, so.
Okay, no problem, no problem. This is why I thought of a beer, but it's fine.
Okay, I hope I can just send you some. All right. Yeah.
We actually have a storm outside, which is very unusual for Israel.
Oh.
Normally it's very hot, but
Wow.
crazy.
I see.
You probably wouldn't call it a storm. It's a little bit of rain, but we call it a storm.
So you don't have so much rain there?
But no, but this has a name. It's a storm that has a name and everything. But it's basically rain. It's
Rain,
not like
I
a
see.
storm in the US. Yeah.
We have a snowstorm.
Yeah, that we don't have. Yeah. But we do have snow in Jerusalem today, which is very unusual also.
I see. Well, so usually this format, I think students start kind of asking the question to you and they freely discuss. So,
Please.
so I think anybody can start. Oh, I think
Yeah, I'll ask questions. So, with the memory part you're talking about, the second half, which is super interesting. So all those sort of memory tests you were doing were basically, they were all with those one same odorant, right?
No, I didn't elaborate.
Okay.
But we checked different odorants, and in fact, there's something interesting there. So we checked, and we found that this is happening exactly as I told you. When it comes to odors that are sensed by the AWC neurons, I don't know if you are a C. elegans researcher or not. No, but C. elegans have a few, not so many sensory neurons that smell appetitive odors. Okay? Two of them that are called AWC, and they sense multiple odors. And we found that this is happening with odors that the extension of memory on ice happens with odors that are sensed by the AWC neuron. However, odors that are sensed by the other pair of sensory neurons which smell appetitive odors, it is called AWA, there it doesn't work. And it's very, very interesting. We even did experiments where we moved the receptor from the AWA where it doesn't work to the AWC that worked, and then it worked. Okay? So there's something specific about the sensory neuron. And this is interesting because the AWC neuron, they also have a completely different architecture. They look like a big Rorschach thing, more affected by temperature, and they also affect temperature, in fact, temperature sensation, high temperatures, unlike the other neurons. So there's something complicated there. To me, it's interesting because it's another proof that it's not simply physics. It's something that happened that has to do with the biology of the neurons, that the different states that it can acquire.
Okay, so that,
yeah.
Yeah, that was like answers one of my other questions, which was, does this work with other, or I don't know, maybe there aren't other tests of memory besides like odorants, but.
This is an excellent question. We didn't do it, and we should, okay? The other things that we can do are to do the same thing on only, instead of volatile odors, to work with tastes like salts and pair them to starvation. This is something that we definitely should do, okay? The other assays are less robust. So there are different types of assays, including assays with where instead of pairing it to something bad, you pair it to something good. This is all, it exists, but it's much less robust. And you know, I don't know how people do behavior in other organisms with super complex, even in C. elegans, where you control their behavior perfectly and everything, you still need a very good assay. So this is why we didn't do it yet, but it would be very interesting to see whether it extends to other assays, and I bet it won't extend to any assay. To some assays maybe, but not to everything.
So I guess I have something that sort of builds on that. I was wondering about other assays that you could use for studying worms. Do you ever introduce like predators to the worms and see how that could, maybe that some would escape predators better?
Right. Excellent idea. This is something that a student in my lab, Dana Bartzvi, is now doing with, she's using Prestioncus pacificus, which is another nematode that predates or eats C. elegans. It's not the most natural thing, perhaps. I don't know if they meet in the wild, but they do try to escape. So it's probably pretty scary for them. And we also thought of using, there's this fungus that chokes the worm. And the worm can try to get away from it. So this is another something that we thought of doing. We didn't do it yet. We tried to keep it as simple as possible. But it's true that these type of things generate very long-lasting memories in other organisms. So it makes sense to try. And maybe these type of memories won't be forgotten after 2 hours. It's possible.
So we talked to, oh, sorry, Elsa.
No worries. I was just wondering, kind of returning to the inherited behavior stuff, how strong those behaviors are. Like, could you overwhelm that inherited behavior with an environmental stimulus, compelling it to not prefer to mate with males or something?
This is a very, very interesting question. I assume that the behavior is, you know, it's within a range, and you can change the range of the behavior by heritable effects. However, we know when it comes to the, and different transgenerational effects will probably behave differently. They will be more or less strong. We know that when it comes to this transgenerational attractiveness, that it survives. I don't know if it's even in the paper, but I remember Itai, the first author, that he did experiments where he mated the worms in the next generation, and it transmits after mating. So at least this type of, you know, drastic thing, it survives. However, we had a paper in eLife maybe last year or 2 years ago, I don't remember, where we've shown that if you start an RNA response by double-strand RNA, silencing a gene, while it typically lasts 3 to 5 generations in regular worms, if you stress the F1s in a number of different ways, you either starve them, put them in high temperatures, or put them in high salt, so it's kind of general, then it shortens the inheritance of the RNA response. So it is plastic, and it can be reset by stress. It's much easier to do when you just, you know, look at the silencing of a GFP or something. When you do a behavioral assay, quantifying it is you know, it's a lot of work and it's difficult. So when it comes to measuring the for how long it lasts and how robust it is, we tend to avoid these experiments, although they are very, very interesting. And it's also interesting to look at the interactions of different heritable effects, so different stresses, and how they are affected by the future or the next generation, what happens to the worms.
Thank you.
I can go next. Do you see any phenotypic changes in the offspring of the worms that were presented into this? I would call them, I don't want, but like the worms that were affected by the research you guys were doing. Do you see any changes in their offspring?
In which offspring of the worms that were, you're talking about the memory assay?
Yeah, the memory assay or any of the worms that you use. Do you see any phenotypic changes? Like, did you see more dumpies in offsprings, or did anything change?
Yeah, so I'm not sure. Everything like that we would have documented, so I'm not sure. It's true that some mutants, for example, definitely have more UNCs, like RD4 mutants, right? And there are trade-offs. There are all kinds of interesting trade-offs. So for example, we had a paper about transgenerational effects to starvation, where the descendants lived longer. However, this sounds like something adaptive, but we also know that it could be because of reduced fertility of changes in developmental timing. And in the end, it manifests in longer lifespan, but it comes at the expense of, there are all kinds of trade-offs that I'm very interested in these trade-offs and studying them. Specifically about unks and dumpies, I'm not sure that's something that comes to mind. I don't remember.
Okay, thank you.
I had two questions. One with the memory, is there something besides cold that can extend their memory? And second, does this give you an idea, maybe you mentioned this in your presentation and missed it, but does this give an idea of how our memory is stored? Like what even is memory physically?
Yeah, so we looked at a few other stresses instead of low temperatures, and they didn't extend memory. We are limited in what we can study, also because we paired the odor to starvation, so that's one type of stress already. But we looked, for example, we kept the worms in liquid, which is a sort of a stress, and we did some other things, and it didn't extend memory, unlike low temperatures. But there could be other things that I don't know. I think there's a strong link to temperature specifically and to cold because of this cold tolerant state that we can switch on and off. So this seems to be something special about the low temperatures and the response to low temperatures and make the worm cold tolerant. But there could be other stresses that we didn't check. With regard, so I was, the second question about what is the memory, I sort of tried to answer it before and say, honestly, that we don't know about. We think that one thing that is changing is indeed deacylglycerol synthesis. And I think, I don't remember the directionality, but I think that when you produce the deacylglycerol, it increases synaptic release. And this is sort of forgetting. So if you delay deacylglycerol and you don't secrete and you delay synaptic release, then you extend the memory. You don't forget. The worms don't respond to the odor. So it could be this, this pathway of deacylglycerol synthesis. That could be in the memory in the AWC, in the sensory neurons. But it could also be something more complex.
There's what is the name of the mutant? Dower mutants, the Dower state.
Mm-hmm.
What happens to Dower mutants in the cold assay?
This I didn't do, but it's a very good question. And the reason is that it's so when the worms are in the Dower state, this alternative developmental state, then they live a lot longer and they need perhaps to remember longer. Interestingly, the neuron that controls the exit from the Dower state is the ASJ neuron. The same neuron that we see regulates the extent that when you disrupt, extends memory maintenance. It's hard to teach worms when they are not adults. So it's hard to do it when they are younger, for example, when they're adults. So we can't do it. We did look at DAF2 or some genes that are related to Dower and examine the effect. It was nothing very simple.
Thank you for the very interesting talk. I also study environmental stress and epigenetic regulation. This is on another model organism, so that was very relevant for me. My first question is just something I want to make clear. What is the parent of origin effect when it comes to small RNA inheritance in C. elegans? And the second question I have is just, what are some of the things to keep in mind when you try to design a transgenerational inheritance assay? Yeah,
excellent question. Which organism do you study?
I do Arabidopsis thaliana.
Great. So, with regard to the first question, parent of origin, yeah. So first of all, we know that both hermaphrodites and males can transmit RNA to the progeny. Okay. If you start an RNA response in males, it doesn't work well, even within the same generation. But if you start an RNA response in a hermaphrodite, isolate males, male progeny, they will carry the response to the next generation after mating. Okay, so it can be transmitted through the male, and there are different small RNAs inherited in sperm and in oocytes, and also different proteins that function in the sperm and oocytes in RNA inheritance. So there are also differences. What we studied about parent of origin is that we saw that if you disrupt the germ granules in the hermaphrodite and then isolate either hermaphrodites or males and carry the response via either the males or the hermaphrodite, then you will see totally different effects. This we published in our Current Biology 2019 paper. The phenotype that we were measuring is disruption of the capacity to respond to RNAi. And it's totally different whether you transmit it through the male lineage or maternal lineage. You get the opposite effect. Oh, the second question was, what is it important to keep in mind? In fact, I'm supposed to write like a methods paper about this for 5 years or something that I'm delaying. But there are many things to keep in mind that people don't think about. And it's very important also for reproducibility. For example, if you go to different C. elegans labs, you see that different labs have different standards for maintaining the worms. In some labs, the worms are totally, contaminated, and they don't care because they do genetics. What do they care? It's genetics, doesn't matter. Or the worms change the temperature that they grow the worms in because they don't want to work in the weekend, so they put them in high temperature, low temperature, so they grow faster or slower. Okay? Or they starve them to synchronize them. All of these things we know lead to transgenerational effects. So you have to be a little bit fanatic. It's borderline impossible, okay? Because for how many generations? Do you keep them well-fed and in a certain environment and uncontaminated for 5 generations, 10 generations? Who knows, okay? These are issues that we're dealing with, okay? There are also, and there are also many things that affect it. For example, C. elegans, the C. elegans strains that were used in the beginning of the C. elegans days, a third of the strains come from a strain that had a mutation in MUT16, which is one of the major RNAi genes that defines the M granule so they're defective, they have problems with RNA. People don't know this. So that's another problem. It's very hard to deal with all these things. You need to try with worms from different labs, keep the worm clean. Another thing that we started to do, which is maybe the ultimate thing, but it has its own limitation, is that we use worms that we created that have the HRD1 protein. This is the argonaut that carry many of the heritable small RNAs tagged with the DAG1. So we can reset and start as a blank slate after inducing of degradation of HRD1 to remove, previous memories. Okay, this leads to its own problems because then how do the worms replenish the depleted pool of heritable small RNAs? Is there some stochasticity in it? Perhaps, we don't know. Okay, so these are problems. So yeah, and I think these are huge problems. In fact, so for example, when people say in C. elegans it's robust enough that you can replicate other people's experiments. But we know that in other organisms, for example, people that work with mice, many people don't believe that it happens. But if you look at the experiments, they're very small. So, they could have all kinds of bottlenecks and the statistics not so good. And also there are genetic differences between the mice. I'm not sure that people even know how to do transgenerational experiments in other organisms. Certainly, I don't think that people know how to do them in humans. So to say that it doesn't happen, or that it happens, what you should say, we have no idea. Okay? And we should think of how to plan these experiments, because if you think about genetics experiments, when it comes to complex traits, we only get a signal when you look at many thousands of people. Right? Now, if you want to do a GWAS, you don't start with less than 20,000 people. Obviously, no one did such an experiment for epigenetic inheritance. People look at 60 people or something. It's not serious. Okay? And even when you look at, and even if you would look at 20,000 people, you would have to somehow control for genetic differences and differences in their environment. It's really difficult. It's a difficult problem. So I just think we don't know how to do it. In C. elegans, we're getting close. In other organisms, not simple at all.
Thank you. Thank you very much. I look forward to that makeup period when it comes out.
Thank you.
I had a question about the sexual attractiveness assay, and I noticed that in the paper it said that sperm had like more defects as part of the response to increasing pheromones. And I was wondering, like, in addition to that, did you see if any of the worms were like actually like dying earlier or like had shortened lifespans, or like I don't know.
Yeah, we didn't measure lifespan. MEG3/4 mutants certainly have a small brood size. That's obvious. Okay. But we also did experiment with many other mutants and also look at the wild type descendants and the responses to temperature and so on. And as I said, I'm sure there are trade-offs. In fact, the trade-offs are the things that interested the most. I think that the environment affects sperm and germline in general. And the trade-offs could be, for example, a change in behavior. And I think this is very interesting. I would be disappointed if there are other things if there aren't any other things that are happening. But we didn't measure enough. There could be other behaviors that are changing as well.
Cool. Thanks.
Thanks for telling this fascinating stories. I have a question about the natural history of worms. So do you think the temperature would co-vary with the amount of contribution from sex selection of worms? Since the temperature does, affect the amount of sex attractiveness that could be generated.
Yeah, could be. I don't know, but it's an interesting hypothesis. It's a good question. But we didn't study this. We didn't do any experiments with wild isolates or something like this. We saw that some wild isolates that are more sensitive to high temperatures indeed generate this transgenerational response. So they're more attractive when they are young. It's not in the paper, but I think it's very, very interesting, and it's sort of the future of this field to look at variation across wild isolates and in different environments. We know very little about their ecology, but that's what gives it the context, and it's very interesting to study. There are very few papers about this in general, about transgeneration inheritance of small RNAs in wild isolates, but there are some interesting works.
Okay. So I have two questions. So first, the question is about the environmental, how the environmental cues sensed by neurons can be transported to germline cells. So you mentioned in your 2019 Cell paper, there are several possibilities. So I was wondering if it is possible that endosomes or other intracellular vesicles have a role to play in the transportation of small RNAs from neuron to germline cells. So that's my first question. My second question is kind of naive. So you mentioned in your first story that any environmental stress-induced phenotype is transmittable for 3 generations. So I was I'm just kind of curious why it's always 3 generations.
Great. So first of all, excellent question. I will answer the second one first, okay? I don't think it's always 3 to 5. I think there are a lot of examples of it, but it doesn't have to be the case. I will start with that, okay? Doesn't have to be the case. Also, when it comes to RNA-induced silencing, it's true that at the population level, the response lasts 3 to 5 generations most of the time. but there are individuals or specific lineages where it lasts a lot longer. And this is something that we studied a little bit in our latest paper. About the first question, how the neurons communicate with the germline. We say, as you said, I don't know if you hear this, but so we said this dramatic answer with the thunders. So we said in our paper very explicitly that we don't know, we can't prove that it's actually the small RNAs that move from the neurons to the germline. We think it's the most parsimonious explanation, but it's very hard to prove. It is possible that there are other molecules like hormones that the small RNA production in neurons affect, and this affects the germline. The way that we go about it, that we try to study it, is we are currently doing an unbiased genetic screen to look for genes that disrupt this communication of the neurons with the germline. So one of our targets, stage 2, we have it tagged with a fluorophore. We have RD4 expressed just in the neurons, and we look for mutants where this communication, this silencing of stage 2 in the germline by neuronal RD4 is disrupted. We hope this will give us some answers or clues, but we don't know at the moment. And it could definitely be, exosomal communication. That's a possibility. But there are but in C. elegans, the neurons coordinate all kinds of effects non-cell autonomously using many messengers, many molecules.
Okay, I'm looking forward to seeing your latest result.
Well, thank you.
Yeah, excuse me. Yeah, I have a similar question with the first question. Do you think there is a kind of screening mechanism for the small RNA when they are transmitted from neurons to germline? I mean, maybe there is a kind of like specific biochemical features for this small RNAs so that they could be transmitted from neuron to germline.
It's an excellent question. It's an excellent question. Indeed, if the worms are secreted through exosomes or vesicles, which is something we don't know, but if it's happening, that there could be something like this. There was a very recent paper about the sorting mechanisms of small RNAs to exosomes. I don't know if you've seen it very recently. And there was also a few papers about a sort of a code. And this is not in worms. This is in other organisms, probably humans, but I'm not sure, or mice, about the sequence features that target certain small RNAs to exosomes. In C. elegans, we have no idea. C. elegans is very good for genetics, less so for biochemistry. So there are very few papers about the composition of vesicles or exosomes in C. elegans. People didn't isolate them and look at the content. Just a little bit of work from Maureen Barr, and that's pretty much it.
So
it would be very interesting to see, but we don't know it yet.
So you don't think C. elegans is a perfect model for the biochemistry?
No. I think it's possible. It's not the perfect model for biochemistry, but people do biochemistry. It's possible. It's possible. There are some limitations. For example, it's amazing, but no one has done a cell line in C. elegans. Okay. There aren't any cell lines. In part because you can't immortalize the cells, and the animals are post-mitotic, so you can't maintain the cells. So, and this is it's a problem for biochemistry, but you can do biochemistry. You get smaller amounts. It's harder, but people do it. But I also think that it's just a matter of tradition that many of the C. elegans researchers are geneticists. But it is possible to do biochemistry.
Okay, thank you.
Hi, excellent talk. Thank you for presenting. I'm not a C. elegans person, but I learned that C. elegans has a lot of different kind of germline granules. And I was wondering if there are any changes in the dynamics between like mRNA, small RNAs, and those granules during inheritance.
Yeah, so the germ granules appear to be more and more important for inheritance. And we found that, and this was also shown by others, that in high temperatures, the composition, the germ granules shrink. Some of the granules, but not all of them. And also the composition of the small RNAs in the germ granules changes dramatically. And this is memorized across generations for multiple generations. I assume this also corresponds to changes in mRNAs in the granules. What appears to be happening is that the mRNAs, as they exit the nucleus, go through the nuclear pore, can get trapped in particular germ granules, and then they serve as a sort of a factory for production of these small RNAs. They use as templates for making small RNAs. And the composition of the RNAs in the germ granules changes transgenerationally. So you are right.
Hi. I had kind of a specific question that I thought of when you were talking about kind of the HSF1 gene and how it kind of results in like the stochastic response of the progeny and kind of epigenetic variability. And it reminded me of other talks I've heard about kind of like lifespan in C. elegans and how there's, despite like a lot of genetic similarities in C. elegans, you can have a very stochastic amount of like how well they live, how long they live, and like a lot of stochasticity and lifespan. I was wondering if you thought there was a connection between kind of that epigenetic variability and that can get produced from the mother, and if that can have an influence on lifespan of the progeny.
Yeah, I totally agree with you. There are these works by Ben Lenner and others about these effects that start with the stochastic gene expression and affect lifespan and other stress resistance and other factors. I think that it could be connected to HSF1 as well. The interesting, or to small RNAs, variability in small RNA functions. The interesting part for me is not only this connection, but it can also be carried transgenerationally. But yes, I think there could be a link between all of these things.
Oh, thanks.
Thank you.
Hello, so I had a question. So with the small RNAs and the progeny that have inherited them, have you done any type of rescue experiments or trying to restore the wild type phenotype in those progeny, or do you have any like ideas for targets that might be available?
Yeah, are you asking about the transgenerational attractiveness?
Yeah. Either one.
Yeah, so when it comes to the effect on chemotaxis in high temperatures, there we did identify a particular target, this gene SAGE2, and we showed that just when we CRISPR it, it explains a lot of the effect. When it comes to the transgenerational attractiveness, we looked at, so we found many small RNAs are changing, small RNAs that affect sperm genes. We looked at the top targets, the ones that change the most in small RNA levels and also in mRNA levels. And we examined individual mutants. They didn't give us the full phenotype. One of them, I don't remember the name now, gave us a partial phenotype, so a partial increase in attractiveness. But we were very limited in what we could do because many of these genes, the mutants are sterile, and then you can't measure them because obviously the sterile mutants will be more attractive. Okay? There could also be combinations of targets that you need to downregulate a few genes, and only in the combination it leads to full attractiveness. My favorite hypothesis is that it's not a particular gene, but instead that different disruption in the levels of different genes that function in the sperm can lead to attractiveness, or perhaps an accumulation of a few of them lead to attractiveness. And I'll tell you why I think this is a more interesting scenario. It's very hard to prove, but I think that it's interesting to think of the sperm as a sort of a stress sensor, that whenever it's messed up because of a different reason, the worms can tell that it's in a dangerous, stressful environment, and it should mate to increase genetic variability. So I think it's an interesting concept, that a sort of a general danger signal. And similar things have been shown in the past, so not with regard to the sperm. By the way, the sperm is sensitive to temperature and to stress in different organisms, so this is a nice hypothesis in general. But people have shown, Gary Ruvkon's lab found, this is a Cell paper from maybe 2012, they found that if you grow worms normally worms like to be on the bacteria that they eat. But if you grow them on bacteria that do RNAi, when the RNAi targets an essential gene in the worms abandon the lawn and they go away from the bacteria. And they do this, according to their hypothesis, I think it's a very nice hypothesis, because they they can't sense any toxin in the world. So what they do is they monitor their own self reactions. And when they feel sick because an important gene is being downregulated, they say, I'm eating something bad, I should go away. So again, this is a sort of a general monitoring of your health and an overall signal of danger that tells you that you should now switch things up. And I think the sperm could be the same way.
Thank you.
Hi, I had a very broad future directions question for you. Based on your talk, half of it was looking into transgenerational inheritance, and the other half was into this memory retention that's codependent. So do you think that in the future you would take the second project and also see if that has a transgenerationally inheritable effect?
Yes, we are definitely looking at it.
Okay.
Yeah, I am, and maybe there will be something. We are now doing it, so I don't even know what the answer is. But we are very interested in doing this. But also, in general, I'm very interested in memory per se, even not transgenerationally. And the type of questions that you were asking, what is the memory? How it is encoded? Can you extend it? So this is equally interesting to me, just like transgenerational effects.
Right, makes sense. And I think she had a question as well.
I was wondering about, you mentioned the HDRE agronaut protein and its importance for its importance in transgenerational inheritance. I was wondering if you've seen any other agronaut proteins like CSR, any of those, have any of those also been implicated in transgenerational inheritance?
Yeah, definitely. So aside from HRD1, there are 3 other argonauts that were shown to be specifically important for heritable responses and not RNA within the same generation. And these are WAGO1, WAGO4, and CSR1. CSR1 is interesting because it doesn't carry silencing responses transgenerationally. Or, it's controversial. Some people say that it does. Some people say that it's the opposite, that it licenses gene expression. But there are also other people that say that it has different roles. It tunes down gene level and perhaps roles in inheritance and roles in early development. So it's so complicated. But there are definitely other argonauts. We were the first to show actually that there could be inheritance, RNA inheritance also in HRD1 mutants. In our 2019 current biology paper, we found that if you disrupt the germ granules, this leads to a change in the pool of heritable small RNAs, and now also HRD1 mutants that typically are inheritance resistant can inherit RNA responses. Okay, there is now a new paper from Sedu, a preprint, it's not published yet, saying the same thing. So, and I think what happens there is that when you don't have HRD1 and you disrupt the germ granules, the wrong small RNAs get made in the germ granules, then they get loaded on other argonauts that can carry transgenerationally. So I think this can happen too.
Okay, that's interesting. Thank you.
Sorry, Angel. I thought you raised the hand first. But thank you, Oded, for the amazing presentation. It was very interesting. I'm recently a C. elegans person, so it was very relevant to my research. And I was just wondering, going back to the low temperature and memory retention, if you had any idea of the gene expression patterns, similarities between lithium chloride and cold temperatures. And I guess my second question would be if you thought about doing the lithium chloride experiments with varying temperatures and what you think would show you about how memories are stored.
Yeah, so excellent questions. We did do experiments with lithium and low temperatures. There is a connection there. Or we didn't do it like this because in low temperatures, they anyway remember longer. What we did is we made the worms cold tolerant and then treated them with lithium, because when they're cold tolerant, cold doesn't extend memory, and it affected the increase of the lithium. So this we did, I didn't show it, but we did it. We didn't look at the transcriptional response to lithium, but it's very interesting to do. And I think that, I mean, if we will find that lithium works through the mechanism that has been described in ASJ, where they found that it inhibits these phosphatides that have another target and so on, then we'll have more about the mechanism. If it ends up not being about this mechanism, then we will look for other lithium functions. Lithium can affect many things. And then it would make a lot of sense to sequence mRNAs from lithium-treated worms, and also look at and also compare it to cold-treated worms. But we didn't do it yet.
Thank you.
Thank you.
Hello. My question is if aging at all affects the ability of the C. elegans to recall memory, like if elderly C. elegans, I mean, I don't know how elderly C. elegans get, but.
Yes, it's a good question. I'm not sure. I'm pretty sure that old worms will remember less well. But there's a good chance someone has done this experiment. It makes sense to do it because it's interesting to look at memory loss in aging. I don't remember a particular reference, but it is interesting to do. The problem is, with every learning and memory essay, you want to start with something very robust. And it can get wrong because of many different reasons. When the worms get old, they are just messed up in general. That's the problem. So to say it's specifically a problem with memory is challenging, but it's very possible that it has been done, and I'm just not an expert at that particular.
Thank you.
But I will be surprised if the old worms remember as well as the young worms.
Hi, I had one more question. So you mentioned that the MEG3/4 mutants with the auxin-inducible degron with the HRDE1 leads to transgenerational inheritance of that premature attractiveness. I don't know if it's really possible to do this experiment in worms, but could you do like a withdrawal assay of auxin and see, if HRD1 is gone for, I don't know, X amount of time and you put it back, does that still lead to the transgenerational inheritance?
Yeah, so we can do this, but HRD1, when you remove it, it's not attractive. It's not on its own attractive. What we've shown is that depletion of HRD1, a temporary depletion of HRD1, exactly like you said, by induction of its degradation with auxin and then removal of auxin, stops the transgenerational attractiveness of the mutants and of high temperatures. But just depleting HRD1 alone doesn't make, or HRD1 mutants are not on their own attractive.
I see. Okay. Thank you.
It resets the memory. Yeah.
Gotcha.
So do you think that there's any maybe even potentially evolutionary advantage of the fact that you see this relation between cold tolerance and increases in memory? Do you think that just happens to be a byproduct of some whatever mechanism is working to lead to that, or do you think there's some sort of reason that those are connected?
Yeah, it's an excellent question. First of all, I'll say that there has to be some trade-off, I think. So it's probably not, why not remember forever? So that's probably trade-off. But we don't know enough about the worm's ecology. I don't think that this is a situation like, with squirrels where they hide their nuts in the winter and then go back to get them or something. I don't think it's something like this. It could be just chance that their circuits are intertwined and connected. It's a possibility. It could also be something more interesting than that. Like, for example, transition between different hosts that grow in different temperatures. Okay? But as I said, we don't know enough about the ecology, but it opens many possibilities. It's even hard to think of experiments of how to test this, but it could be.
Yeah, probably knowing more information about other like odorants or other memory tests would help inform that.
Right, and we're also examining wild isolates that are more or less called resistant to see whether this is whether the links are conserved. Yeah.
Following Myron's question, I was just wondering about memory. Is there like some traits of memory that are related to memory that we know that would increase fitness of worms?
Some memory that would increase fitness. Some memories that are worth remembering because they actually will save you.
Yeah.
Yeah.
Like those any memory-related traits that might, help worms survive.
I'm trying to think. So one thing is that the worms can remember the cultivation temperature, and then if you do a temperature gradient, they will, when they're older, go back to the temperature where they were fed. Again, we don't know, we don't have this in the wild or in a natural setting, but this sounds very adaptive. But the links to ecology are very limited.
Sweet, thank you.
I bet it's helpful, but the connection to a real life situation, elegans, people are getting into that, studying worms in the wild and looking at wild isolates and their evolution and so on in the wild, in natural settings, but it's just the beginning. And it's complicated to do.
Yeah, I'm asking this question because I'm studying Drosophilus, and my next project might be comparing Drosophilus from different thermal conditions. So I'm asking this because I'm sorry, with the designing trait that I could expect that there might be changes in fitness due to difference, in those phenotypes related to memory.
Right.
So I have a broad question. I do work on memory in Drosophila. So one of the things that strikes me is you clearly contrasted the conditioning that generates short-term memory versus these other training conditions which generate transgenerational memory. So you've got the two extremes, the one that is producing short-lived and ones that are producing very long-lived memories. So the question is it that the long-lived, the stimuli that you're using in the long-lived are much more life, death, sex, or starvation, right? I mean, it's kind of like, and the way that the Bargman people did the chemotax, the plate chemotax, the one with that it's less. And this relates to Suyan's question that just came, right? It's just sort of like how salient for survival and for producing progeny, is that what's different about these cases?
Yeah, it's an excellent question. I think, in my learning and memory within the same generation, we also starve the worms. So, but we starve them for 90 minutes. And in the transgenerational essay, we starve them for 6 days. So it's true that it's more severe. But these are also experimental limitations. But your question is very relevant and it makes sense. Perhaps we can look at it from a different direction, that worms, because they have such a short lifespan, and also perhaps because they are genetically identical almost to one another, they think transgenerationally, it's like the same organisms. And also, they only live for a few weeks, but the next generation will be there in 3 days. So the chance that the environment of the parent will resemble the environment of the offspring is relatively high.
Yeah, yeah.
But in 3 weeks, it's like 3 generations. No, it's or more. So perhaps that's another way to look at it.
Yeah. Another sort of from a behavioral perspective, you know, I think one of the characteristics people think about for a behavior is extinction, right? So you give them the same stimulus without either the reward or the punishment that you train them with. And does that decrease, if you give that repeatedly, does it decrease their response, right? And again, I'm thinking you have certain behaviors which really look like life or death or breeding. And but they're not like calculus, right? I mean, it doesn't matter. It doesn't matter if you can integrate, right?
Right.
And so if you took, can you extinguish the transgenerational thing? I guess that would be one.
Yeah, we can extinguish it by stress.
Oh, the same stress without the.
No, so actually.
Different stress.
No, yeah, so actually, yeah, same stress accumulates. So for example, if you grow worms in high temperatures for 1 generation,
yeah,
or for 5 generations, the intensity of the transgenerational response changes. It becomes stronger if it's multiple generations, but it's always a stress. It's not an extinction of the stress.
Okay. Okay.
So you can look at it the other way around. That when you don't stress them, you extinct the stress. And we also showed that if you do RNAi, then you get a transgenerational response of 3 to 5 generations. But if you do RNAi again in the F1, it extends the ancestral response, even if you do RNA against a different gene. Just the activation of the external RNA system.
So that's the counting. There's some kind of counting.
Yeah, or accumulating counting. Yeah.
Yeah, it's additive. Yeah. Okay. Yeah. Yeah. Anyway, those are really cool experiments. Thank
you.
The transgenerational things are very cool.
Thank you.
Yeah. We have other questions. How are we doing? We're pushing in towards 1 o'clock, you know.
I'll throw one other quick question. So with the again on the short-term memory and the cold temperature, can you may be limited in the system in this, but can you do anything in the opposite direction, like seeing if like higher temperatures shorten the memory?
Yeah, we can, I think. We didn't do it, but we did check whether AFD mutants affect it. AFD affects sensation of high temperatures. And it did have some effect. It wasn't a full effect, but it did affect it.
You know, just to add one thing, there's a phase of memory in Drosophila when you pair one of 2 odors with electric shock, so negatively reinforced. That's called anesthesia sensitive memory, anesthesia resistant memory. And it's basically train the flies and then put them on ice.
And it prevents memory, right? It's the opposite.
And no, it prevents. It's a complicated thing. I was trying to think through this when you're talking. There's a component that is inhibited. So there's a part but then there's a component that's resistant, that is not, that is left. And I think the field now, and there was a lot of confusion because people thought those 2 components were totally independent. I think the field now is more moving towards, there's some complex relationship between those 2 memories that we don't understand. Okay. So yeah, and my personal feeling is that one might be converted to the other. Okay, that it's actually the anesthesia, it's kind of a state where the animal is, you know, the brain is assessing whether anything new information should inform it to forget the older information or should inform it to preserve the older information, right? I think it's along those lines that these are
interesting, very interesting.
So that's very interesting. The anesthesia, the cold thing is very interesting. That's very interesting. I'll send you something on that. I'll send you.
Thank you very much.
Yeah. Okay, anybody else have questions? That was great. We had a ton of questions.
Excellent questions.
Ask a question.
Question is about the number of generations treated by the heat shock, because you told us it's a kind of accumulation of memory. So do you think it's a kind of in density dependent of the small RNA? Maybe there is a kind of threshold of the small RNA when the density of small RNA reach this threshold, it could be maybe inherited by the progenies.
Could be. We honestly don't know, but it could be.
So you don't test the density of small RNA.
The problem is we don't know anything about the stoichiometry. How many small RNAs per mRNA? Because we can't sequence small RNAs from a single cell.
Oh.
You can do mRNA, but you can't do small RNAs. We're trying to do it now to understand in a given cell, in a fertilized egg, how many small RNAs do we have per mRNA? This is something that we're trying to do now. It's at the limits of the technology. It's not really possible. You can do mRNA sequencing, of course, but not small RNAs, but we are trying to assess it in a number of ways.
Okay, thank you.
But it's very important. The stoichiometry will tell us a lot.
All right. I think we should give Oded a huge round of. Well, next time you come to Madison, we'll buy you a drink, but we can't get you.
Thank you
tonight.
Thank you very much.
And you can talk in the daytime.
Yeah. Yeah. Thank you so
very much.
Yeah.
Thank you. Thanks a lot.
Thank you.
It was very good.
Thank you.
Bye-bye.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Thanks.
Thank you.